Welcome to Berries in the Interim podcast, where we explore the cutting edge of innovative clinical trial design for the pharmaceutical and medical industries, and so much more. Let's dive in.
Hey, welcome everybody back to In the Interim, where we live in the interim analysis of clinical trials, the interim analysis of science. And I'm joined today by what has become now your fourth or fifth time on In the Interim. Kert Viele has joined me, and Melanie Quintana, who leads our consulting group here at Berry Consultants. And I'm going to lose control of this podcast today, and I'm going to hand it over to be the host to Kert. And Kert, you want to be the host and introduce the topic for today?
Yeah. So anyway, I like being in charge. I like interrogating Scott. So the topic today is something that we probably should have talked about a while back, given what we do day to day, which is Bayesian statistics and how it's relevant to clinical trials. We want to talk about what it is. We want to talk about when it's useful. We want to talk about standards of evidence and how it interacts with adaptive trials. We're going to hit a lot of stuff. So let me start with the absolute hardest question on the list. And for Scott, can you define Bayesian statistics?
Wow. Maybe I shouldn't have let you be the host.
I know.
If you're going to do this, if you're going to do this to me. So we can do this mathematically, and I don't think people are interested in mathematically, where we have a prior distribution in our parameters of interest. We do create a model like frequentist statistics, but we have a model for the data given those various parameters. And conditional on the data, we update our uncertainty about the unknown parameters in our model. The beauty of that is the prior can be a wide range of things. It can be external data, it can be prior opinions on that, it can be hierarchical structure, but and within the context, it's based on our current knowledge of data, what's our current posterior distribution, what's our current uncertainty about the parameters of interest.
All right. Can you give, so I'll turn this one over to Melanie. We've often talked about this. This comes up in a lot of regulatory documents and various other things as the use of informative priors. Somehow I want to say, hey, I already know something about what the parameter is like. And we'll certainly get to that as we go, but it's also a lot broader. And Melanie, can you give us some examples of places you would see Bayesian analysis in a clinical trial in addition to borrowing historical or external data?
Yeah, I would say actually most of the trials where I've done a Bayesian analysis, for instance, as like the primary analysis or Bayesian analysis within the trial itself, most of them actually have non-informative or vaguely informative priors. So I actually rarely do it in the space of this. I have this really informative prior and I want to use it. I think it works great in the situation of we have other evidence that we want to synthesize within the trial itself or slightly adjacent to the trial. And we want to use that evidence to sort of come up with an overall synergy of information that we can then use to estimate our current parameters. So I think there's lots of examples. For instance, borrowing information across concurrent and non-concurrent controls in a platform trial or something like that. That's not using an informative prior, that's borrowing information across multiple sources.
So with respect to that, we've talked about the modeling aspects of Bayesian. We also use Bayesian a ton in adaptive trials. Scott, could you talk about why we might use it in adaptive trials? Why is it a natural fit there?
Yeah, I tried poorly probably to describe Bayesian. One of the nice aspects of it is you start with a distribution of the parameters, you observe some data, and that's updated based on your current information. And by the way, that could be auxiliary markers, early clinical markers, a 30-day outcome where 90 days is primary. And now you get an updated distribution of the parameters of interest. And if you collect a little bit more data, it's updated again. And you get the same answer if you update 5 times or if you just did it all in one fell swoop. So it's this beautiful learning mechanism based on the current level of data that captures the appropriate amount of uncertainty. So if I'm going to make an adaptation in a trial, I'm going to select a dose in a seamless trial. I'm going to make a decision on enrichment. Do we want to drop a population or not? Based on all the data I've collected in the trial, what is my uncertainty about these parameters, about the effect in this population, about this dose, the likelihood of success in the trial? The Bayesian approach is such a natural and beautiful way to do that. Part of it is also the real awkwardness of using frequentist statistics in that paradigm where analyses are based on the sample space. And in adaptive trials, that gets really, really complicated very, very quickly. And in the Bayesian approach, it's a very natural thing because you only have to analyze the data you've seen and not concerned about data you haven't seen, which does matter in a frequentist analysis. So it's a very, very natural way to continually update the distribution and then drive adaptive decisions. Even if at the end of the day, the trial's analyzed using a p-value, you can design better, smarter trials by using Bayesian throughout the adaptation process.
So you talked about, kind of harming on the last word that you'd said there, which was a better trial. I've often heard this notion that if you're using Bayesian, and really adaptive in general, that it's a shortcut. The goal is to do something smaller, to try to make decisions on less information. If I use an informative prior, I need less data in my current trial. I'm trying to make the sample size smaller. A lot of times it's talked about less information, that you're trying to, usually it's referred to as lowering the bar. So, are Bayesians lowering the bar, or is there more to it than that?
Yeah, it's absolutely not. Now, in some circumstances, there's a Bayesian analysis that's walking in that could have aspects of lowering the bar. Depending on what that means, it could be the success criteria is easier than a traditional 5% two-sided type 1 error. It in a scenario could end up being a smaller sample sizes. But many times by employing these methods, we're actually collecting more data in the scenario. We're analyzing more clinical outcomes combined together. We're borrowing from a patient population in that scenario. We may be utilizing more data to drive the answers. If you're running a trial where you're estimating the effect in pediatrics and you're borrowing from adults, you're using more data to provide a better answer of pediatrics. So there's many circumstances, both in Bayesian and in adaptive, where we actually go bigger and we collect more data. We have bigger phase 2 parts in the phase 3 trial. So I think it's actually in many ways kind of the opposite of that, but yet I think we're labeled as that.
Yeah, I think that's right. I think in Bayesian settings, we're trying to efficiently use all of the information in front of us many times. And because we're sometimes bringing in more information that otherwise you might have ignored, in some senses, we might be raising the bar. Like Scott talked about multiple endpoints. You might have a model where we're looking at what's the common treatment effect across multiple endpoints now. And actually, then now we might have to raise the bar a little bit. We might have to show that each of these endpoints are all significant to actually be positive. So in some senses, certainly, I think we could possibly be raising the bar because we're bringing in more data.
So Melanie, you're involved in the HEALEY ALS Platform Trial, which you can go back to a previous podcast with Merit Sokovich and go into that. But you have a Bayesian analysis at the end of that trial. And why Bayesian in that trial? And it combines sort of multiple things together, hopefully for a better answer.
Yeah. So that's a case where we want to be able to, in a platform trial, share controls across multiple treatments that are being tested. So each treatment might have its matched controls, and we're sharing controls across them. The Bayesian setting lets us kind of borrow information across these different sets of controls, but not pool the information. If the controls are looking very different, we might share more across and borrow more information if they're, less information. If they're looking very similar, we might borrow more information. So the Bayesian model really lets us get at that like dynamic borrowing of the shared controls. It's really this thought that just because something might be slightly different, we don't need to throw away the data because it might be a little bit different. We can model those differences and build in and use all of the data to get our answer. So that's one of the reasons we're using a Bayesian model in the ALS platform trial. And I think often why we use Bayesian models in platform trials. In ALS in particular, there's this unique situation actually in multiple neurodegenerative diseases. Now we're seeing fatal neurodegenerative diseases. We're seeing these models that are joint models of a functional outcome and death, time to death. And so we want to be able to capture what the treatment effect across both of these endpoints and get an integrated assessment of how the treatment is affecting both function and time to death. And again, the Bayesian framework is well suited to get the sort of integrated assessments across multiple endpoints. So that's another reason why we're using the Bayesian model and why we might use it even in a non-platform trial in ALS to do this joint modeling of multiple outcomes.
So related to that, could you have been a frequentist and accomplished the same goals? I'm going through my hits I'm going through my hits here.
Sure. No, certainly. I mean, I think you could. Would it have been as easy to accomplish those goals? I think you could build to share the controls across multiple what we call regimens in a platform trial. You could build sort of a frequentist meta-analytic model that might be able to do that. I think it gets a bit more difficult. In the Bayesian setting, it's just very natural to build these hierarchical models that can really effortlessly borrow across multiple sources. So yes, I think you could do it. It would have been, at least for me, a little bit less natural to build a model. The same goes, there's certainly frequentist model, joint models that model both function and time to death. Again, it can be a little bit less natural, at least for me, to build those models to get them to do the exact thing that we want them to do, in particular, to have this integrated assessment across all of the endpoints. But there's small features, I think, that are a Bayesian Bayesian framework might be a little bit better suited for, or at least a little bit more natural to build the model in that framework. But sure, you could do something similar in a frequentist setting, I think. One could do something
picky
similar.
A little bit.
I don't know if I could. Someone could.
I'm being picky, but part of the idea here is that Bayesians are naturally trained on this idea of collect data, evaluate, collect more data, evaluate, collect more data, evaluate, which is essentially the adaptive trial formulation. It's actually the drug development formulation. We just put them in separate trials. So this is a very natural setting. I think there are situations where there are no frequentist analogs. Notions like predictive probabilities, so the idea of predicting the next trial or just the completion of the current trial, you have to take into account the uncertainty and the actual, what is the treatment effect? And conditional power, it's close, but conditional power has to make an assumption on what is the treatment effect and project forward. Whereas, the Bayesian idea is all these things are unknown and they're all being used, in a synthesized, coherent way, which I think is very, very natural in this setting.
I mean, in some ways, Bayesian is easier. Conditional power works if you have enough data to estimate the effect. Well, but you have to be smart enough to know what is that sample size when we can use it. A predictive probability just works. Based on the distribution, you update it, you can calculate what's the predictive probability with the current sample size will be successful with a larger sample size. And it incorporates uncertainty in the right way that you can be really comfortable that that's going to give you the right answer. Where conditional probability sometimes works, but sometimes it gives you really bad answers because it doesn't provide uncertainty at the current place you're sitting at.
At one point in we'll call them the old days, we took a Bayesian design and were asked to make it frequentist. And we went back through and converted all of the posterior probability thresholds to p-values and essentially replicated the design. But one of the fundamental issues is we would have never found that design or naturally came to it without looking at it in a Bayesian framework. So we could translate it, but we wouldn't naturally think of it from first principles.
Yep.
Speaking of regulators and old times and new times, where is the regulatory picture for Bayes? Where has it been and where is it going?
Yeah, it's where has it been and where is it going? We're coming out of, you said the olden days. Barry Consultants has been around for 25 years. I've been doing this 25 years. Don has been doing this much longer than that. 25 years ago, there was very rare to see Bayes, especially in an FDA trial. In the scenario, I think the first FDA approval at CEDAR was like 2008, 9, 10, something like that. Very little mention in guidance documents, no mention in ICH guidance documents. Now we're at a situation where CDRH has a guidance document out. We have guidance documents out from US FDA. We're supposed to be getting one from the EMA, I think within a month, a guidance document out on Bayes. The draft guidance that just came out on ICH-E20 mentions Bayesian designs. They're being used in confirmatory trials. They're being used a great deal in learning trials. Outside of the FDA, they're being used in comparative effectiveness. They're being used in NIH trials. It's being used a great deal, being accepted by FDA and EMA, and is part of the guidances for adequate and well-controlled trials.
Yeah, and I'll say too, the whole innovative design program at the FDA, many of those trials that were designed were used Bayesian in some way. Many of them borrowed from external data. So I think that really helped a lot with the getting more familiar Bayesian designs in front of the FDA, knowing, what sort of simulations we might need to show them to get them comfortable with the analyses and those types of designs. So I think that really helped move things forward as well.
So an interesting question, Kert, might be, what use is there of frequentist statistics in clinical trials? And I say that only from the sort of perspective that I do think in almost all of science, the perfect scenario for a p-value, and maybe the only scenario left in science where people think it's relevant, is a phase 3 clinical trial. And in part, it's because it's so prospectively defined. In every other scientific field, there's p-hacking going on. There's sort of abusing p-values or misrepresenting them. And Bayesian's making huge inroad and all of that. In clinical trials, that's still, if somebody came to me and wanted to run a fixed trial of a pretty standard endpoint, phase 3 trial, it's one of 2 adequate and well-controlled trials, I'd tell them not to do Bayes. If there's no reason for that, if you're not bringing in the extra information, you're fighting an uphill battle within that. I mean, that's sort of the place that in that scenario. Now, if you're talking about a rare disease, if we're talking about disease progression models, ALS modeling multiple inputs, you get out of that mold of where frequentist hypothesis testing, it's really the last part left in clinical trials of that. And as you move off of that to learning trials, to adaptive trials, to comparative effectiveness trials, all of this, it kind of loses any kind of benefits, and Bayesian has a good bit of value to all of those scenarios.
So you got a little too close to my pet peeve, Scott, so I'm going to mention it. So there's a kind of a closet industry right now where I run a regular frequentist trial. I want P less than 0.025, and I get P equal 0.04, 0.07 one-sided, so it's not significant. And then what I do is the following. I go and write a paper, and I look at priors, and I come back and go, okay, now the posterior probability of superiority is 93, 94, 95%. And now I want approval. The Bayesian design, it's often presented as, I've now added something that has made this approvable, whereas the frequentist design didn't bring it out. So I know that I have two strong opinions. I'm going to let you guys take a shot at it. So is this a good thing for the world, a bad thing for the world?
Mixed. We being Bayesian, it's not uncommon that somebody runs a failed trial and, let's call the Bayesians. Maybe they'll give us a different answer in that scenario. I think you're largely describing this scenario where we would probably be on the side of regulators that this is not approvable in that scenario. Now, there are surely some scenarios where what bothers me is analyzing a trial as black and white, success or failure, and that's it. What would be really interesting for your scenario is, okay, suppose it's not approvable. What does my next trial look like? Do I need a whole new trial of a bigger sample size to get below, or could I prospectively use that in a new trial? But I agree with you, this sort of last grab of cherry-picked information to recreate a prior and recreate and show success, we think is bad science.
And you bring out, related to this discussion of lowering the bar earlier, we talked about this trial. We wouldn't be saying the trial's good enough in and of itself, but we'd be saying that potentially you could combine the information that you currently have with new prospective information, and the totality of the evidence may reach or exceed the bar that you want in the first place. So the goal is to essentially use the Bayesian methodology to reach the high bar, just not with one trial, as the case may be.
Yep.
So Melanie, do you have examples where we've done this?
Put together the totality of the evidence.
Yep.
Prospectively or retrospectively?
Both, whichever comes to mind.
That's a good question. Prospectively, I think Scott is a good example, actually, of an FDA approval. Yeah.
I'll give Melanie time to think of other scenarios, but yeah, we have had scenarios in really exactly in this case where REBYOTA is an FDA-approved biologic for the treatment of a rare disease, C. difficile infection, recurrent C. diff infection. And the primary analysis at the end of the phase 3 trial was prospectively to combine together phase 2 and phase 3. It's really interesting. You go to the advisory committee meeting on the discussion of this, but a Bayesian analysis combined together both of those and ended up jumping a 99% probability hurdle, combining both of those together. Yes, it was a rare disease. There are particular circumstances about the inability to enroll these patients in this scenario. So there was a need to do something better. in the scenario. If you go to the FDA label for REBYOTA, it's a fairing product, there are only posterior probabilities. There are no p-values in the label. We have some device examples where this has been done, numerous examples. We're actually working on some additionally. And you can imagine the scenario, being a regulator, suppose you're a regulator and somebody comes to you with a trial design, it's borderline, and you actually probably debate long and hard amongst the people at the regulators, and it's just not there. It's close, but it's just not there. Do you tell the sponsor that it's as though you're at time zero? That you have no credit for this, even though we think it's close and the treatment works, but we can't approve it yet? Or should the next trial be different? Should it include the information? Should this be incorporated? I think it has to be incorporated. So those types of scenarios are not uncommon, where you're trying to utilize all of the information to make better answers.
Yeah, and I think Scott's kind of talking about the end goal, and the best example to show that Bayesian can help us get across the finish line for approval. But if you shift more to even like early stage drug development, I think Bayesian models can really help us determine if you should carry a product forward or not. So there's many examples where we'll take, maybe in a rare disease with multiple endpoints, we'll synthesize sort of all of the available information and get a good sense of, does this treatment actually work or not? We'll show that to the company. It'll help them make a phase 1-2 decision or a 2-3 decision. Oftentimes, if the company is needing investments, we'll do those analyses so that investors can get a sense of the totality of the evidence. So it's a great need for using Bayesian in the early phase development to really synthesize all of the available information and make a go, no-go decision, or show the information to investors, I would say.
So you act as though you have one pet peeve, Kert. Do you only have one pet peeve?
I only have one pet
multiple?
peeve for this podcast. I try to get one out every podcast. I'm saving all the rest.
One per podcast. Okay, so. One per podcast. Yep. Can I throw out a really cool application of Bayes? So I maybe I'll save that example for a whole podcast on the topic.
So you have one good example per podcast?
Yeah, that's all I can do. Every time I think of a good idea, I do a podcast. So I have one per podcast. I think the world is going to where we have a much better understanding of disease. And it's not that there's one big disease in the scenario. And so we're going to be running more and more trials where there are subgroups of disease. That we run a trial and there are 5 subgroups of disease that we recognize are slightly different, but they're related. It used to be 10 years ago, we would have run that trial with all 5 groups in one trial. And maybe the treatment failed because of heterogeneity over the treatment thing. That we just didn't understand the disease well enough in that. We used to enroll Alzheimer's trials with people that didn't have Alzheimer's. We didn't understand the disease well enough in that. So now if I'm running a trial with those 5 groups, do I do 5 separate analyses? It's almost within the frequentist framework. I have 2 choices. I do independent analyses of the 5, or I pool them all. But a Bayesian analysis of that, where you're estimating the effect in group A based on the results in all 5 groups, is just better answers. And this dimensionality of disease is going to be a huge problem for frequentist statistics. It's a very natural thing in Bayes, and it's a huge problem for us as scientists, but I think it's an area that Bayes shines. And as this goes forward, this, by the way, is completely not lowering the bar. And it's a case where it prevents we didn't work in 4 of them, but we look kind of good in the 5th. Bayes largely would say that's probably doesn't work at all by modeling all of them. And so it's not lowering the bar at all. It's actually better answers.
Okay, so I have a second pet peeve for today.
Nice.
I do. So anyway, so occasionally this idea we get pushback on this. The idea of we don't want to borrow, there's often a prior involved, we don't know what the strength of it is. We get questions such as, if you're going to ask 5 questions, you have 5 subgroups. Okay, well now is there a multiplicity involved? You really get down into the weeds of this. Where, as you said, these are the kind of mistakes we were making all the time before. If we approved a drug for all 5 groups in a pooled analysis, presumably it doesn't work for everybody in that trial. We just didn't look. So I haven't even hit my pet peeve yet. But anyway, one of my pet peeves, though, is a lot of times this kind of analysis got done after the trial. So it was done post hoc. I'm going to look at subgroups, and it's not pre-specified. There's not a lot of standards to it. And I always found it frustrating where to propose it as a pre-specified design and say, hey, I'm trying to control the error rates, I'm trying to make good decisions, that there's resistance, but then I can do whatever I want on the back end. It led me on occasion to tell clients, you're better off proposing the pooled analysis and all this stuff just doing as exploratory later, which kills my soul in terms of, and I think we're much better off now. I think there's been leaps and bounds. The FDA has a great use case on their website in doing this. But anyway, I think you're exactly right. The more we have subgroups. Let me ask you a follow-up on that related to hierarchical modeling and everything related. Aaron Judge, we talked about his batting average in May at 400. We said we were using prior information, and he was the highest of a group of a number of players. What's his batting average now?
I don't know the answer to that. I should have prepared for this, but I will guess 340.
It's a little higher.
Okay.
So I haven't looked for a couple weeks, but it was 360 something last time I looked.
Okay. Okay. Yep. So he is regressing to the mean.
He's regressing to the mean. We're not quite sure where his mean is. I think Nick said somewhere around 330 he was expecting.
Okay.
So we'll see.
So disease progression modeling, Melanie, you've done a lot of rare diseases and utilize Bayesian progression modeling.
Yeah. Talking about pet peeves, you guys were making me think of mine. My pet peeve, it's funny, you said you're stuck then to choose, are you going to pool or are you going to just test within the individual? And I think a lot of times in progressive disease, the choice then, if you pool, you have no power because it's so heterogeneous and some of the people have totally progressed on the endpoint you're looking at, and so it's hopeless. So what people do is they enroll this very narrow subset of the population that they think is homogeneous and fastly progressing, but not necessarily the narrow range of the population that the treatment might work in, and they're making rare diseases even more rare by only enrolling in a small subset of the population. So yeah, disease progression models are one thing that we do a lot in these rare diseases. I think one of my favorite use cases to date of Bayesian analyses is within a disease called GNE myopathy. So this is a disease that's a slowly progressive muscle wasting disorder and kind of starts in your lower limbs and works its way up and out of the body. I think the investigators at the NIH came to us, and one of the endpoints was the 6-minute walk test, if I remember correctly. God, I think it was. And there are many people who are so advanced in the disease that can't even walk anymore. So it's just, that endpoint is completely hopeless. And this was, I don't know, I want to say one of my first projects at Berry, so now maybe like 12 years ago. Worked with them to develop a disease progression model looking at muscle strength across multiple endpoints. The primary analysis is going to look at the overall slowing in disease progression across multiple endpoints. So if it's somebody who's non-ambulatory, it might look more and be more informed by the muscle strength on their upper limb. If it's somebody who's just starting to progress, it might be informed more by the lower limbs. So it really kind of does exactly what we would want it to do, and that the treatment effect is informed by where you are in the disease. So yeah, that's one of my favorite uses, I think, of a Bayesian model. And that trial, even though it's designed, I want to say almost 12 years ago, is going to read out. We're going to hear about this trial. So we'll have a podcast on it. I think probably in the end of this year, we're actually running that model. The trial ran, and that's super exciting. So it's a great use case of using all of the information that you have as efficiently as possible. We use all of the endpoints so we can enroll a broad population, and hopefully a good use case for Bayesian. Hopefully the treatment works, or at least we get a definitive answer of if it does or doesn't work.
So fully Bayesian primary analysis based on a disease progression model. I also think that's where I talked about the subgroups, many subgroups. We're learning much more about the heterogeneity of diseases. We're also going to be stuck with much more much better endpoints in, think about wearables in a scenario, think about the complexity of the endpoints. I think we use a lot of very, very dull instruments in clinical trials to identify whether treatments have an effect. And we have a great deal of ability to use multiple endpoints simultaneously for understanding effect. Imagine a trial where a single endpoint shows an effect and the other one's secondaries don't. Now you're sort of stuck with, the totality of evidence, what it means. But maybe in the integrated analyses or combining together multiple endpoints, much like your trial does, is going to be hugely more valuable in incorporating, the totality of evidence.
And by the way, Aaron Judge, 349.
349, regressing as we speak.
All right, Kert, I'm going to turn it on you. What's your favorite Bayes use case in a clinical trial?
My favorite Bayes use case in a clinical trial. So I do borrowing. So that's definitely all of my use cases refer to borrowing. We've done actually kind of a regret, a trial that never actually ran. We had a wonderful experience designing an antibiotic trial, which talked about external evidence and also talked about borrowing between you can have an infection at different sites in the body. So you can have a UTI, you can have a lung infection, an abdominal infection, a skin infection. And we talked about borrowing amongst those. And it had some interesting aspects because a lot of antibiotics, if they don't work in a specific part of the body, it's probably the lung, because that's one of the hardest places to get to. So we were talking about ways to actually let the lungs split apart from that in the modeling. But that trial, we had great interactions with FDA. It was part of a grant that actually involved FDA review, but it did not get off the ground. As it turned out, it had funding issues after some changes in funder priorities. But that would have been my favorite.
What about the ROAR trial? I would have guessed the ROAR trial.
So ROAR is a fascinating story. So ROAR it's a basket trial, meaning they looked at a drug in multiple rare cancers, and it had borrowing across the subgroups. Certainly I would call that my most successful Bayesian trial. Certainly multiple approvals. This was the first drug that got what's called a PAN approval. So it's approved for any cancer that has a particular biomarker. So at some level, it's a pass setter in that. The interesting thing about that trial is the drug it's one of those trials where the drug is so awesome that any analysis would have gotten the same answer. So there is that aspect to it where I can't claim that the Bayesian put it over the top, so to speak.
Yeah.
Speaking
think
of
we're
any
running into-
analysis would have got the same answer, I have another question. How do you feel about mixing Bayesian and frequentist analyses? That answer.
So can you give an example
you
of
know-
what would be mixing?
You mean at the end of the trial that some of them are interested? So okay. Yep. So, but what's clear is we need to make Melanie the host next time.
Yeah.
She asks better questions, Kert.
Sorry.
So part of this is probably a reaction. The New England Journal of Medicine has multiple times to us come back and said, if the primary analysis is Bayesian, everything is Bayesian. And I don't care if you set it up in this app to do it one way, we're going to make the whole thing Bayesian. So we used to do some of them Bayesian, but not all of them Bayesian. Now, in circumstances where this could potentially be there, we've created the entire trial from a Bayesian approach. So I like it. I like that if you're going to do it Bayesian, let's do it all Bayesian in that circumstance. There are certainly scenarios where mixing them and providing both have benefit to understanding what's going on. So I don't take a high road that, the two of them can't exist within a similar report, that there's not value in both.
So I don't know what, I certainly, I agree with you. I like that they all Bayesian if you can do it. I don't know that this is solving, to forbid the mixing. I don't know that it's solving a problem. You certainly want to pre-specify. I don't want to do, hey, I'm going to do the Bayesian, I'm going to do the frequentist, and I'll report whichever is higher. That's not, but you end up in a case where different people are doing the primary as opposed to all the safety analyses. They have different expertise. I don't know that, anyway, adding a ton of extra work for what feels like an aesthetic choice. Anyway, I'm not going to, it feels unnecessary to me to worry about it.
Yeah, I think it can be really helpful sometimes if you have a complex Bayesian model to show studying more simple, traditional frequentist summary to maybe the complex Bayesian model, maybe this GME myopathy model feels really black box and complex and people don't know what's happening. So can I pull apart the pieces and show some simple frequentist summaries that people can say, hey, okay, maybe the complex Bayesian model, it has a lot greater precision. We get a strong posterior probability. Maybe we're seeing directional effects from these frequentist results, but that's making me believe your complex Bayesian model. So I have no problem with mixing them. I think sometimes it's nice to show that you get at least not completely different results with the two types of analyses.
All right, so we're being Bayesian. We're in the interim, and we will pick this up. This conversation will continue as we talk about Bayesian, non-Bayesian trials. We bring you examples. But today we were in the interim talking about Bayesian. Kert, thanks for hosting. Melanie, thanks for joining us and taking over the host role for Kert. That was wonderful.
Thanks.
And thank you all. Till the next time, we are here in the interim.